Hi everybody, today we will be talking about COVID vaccine dosage interval. I am Dr. A. Padmanabhan, and what pushed me to take up this topic is the central government of India made the following announcement a few days back on the 15th May. It has come in the paper, in the print. Based on available real life evidence, particularly from United Kingdom, the Covid-19 working group has agreed to increase the dosing interval between the two doses of CoviShield to 12 to 16 weeks. No change in interval for Covaxin was recommended. Now they went on to add the decision to delay the second dose of Covishield vaccine up to 16 weeks was not due to shortage of vaccine, what was based on scientific evidence from the UK. And it was taken by consensus of a group of experts. Now, we all know there is a shortage of vaccine, and when such a decision is made, even among the doctors, there was a lingering doubt. Is it based on real science? Is it based on real life evidence? People would like to know what that real life evidence is. And is it okay to postpone it 12 to 16 weeks? To add more concerns to the public, the British government within 48 hours on Friday decided to shorten the window between the first and second dose of COVID vaccine. That is, they are giving AstraZeneca, which is CoviShield, and brought forward the second dose from about 12 weeks of the first dose to 8 weeks of the same time, 8 weeks. This was done to address the concerns about the spread of B1617 variant, the virus that originated in India. That is of greater concern to India. Therefore, we seem to be going in one direction. They seem to be going in the opposite direction. Announcing the decision to reduce the interval between vaccine doses, the British Prime Minister Johnson said, "We have a larger cluster of B1617 variant, first observed in India since last Monday. Believe this variant is more transmissible than the previous one, but we don't know by how much." Therefore, naturally, how can two governments take diametrically opposite views based on hard science? That's what we would like to look into today. We'll try to answer some fundamental questions today. Number one, the most important, probably the central question is, why is India increasing the dosage interval to 12 to 16 weeks for Covishield? What's the scientific basis for that? And while UK is shortening the interval, citing similar threat to 8 weeks. Why is Covaxin continued to be given in 4 weeks interval? And then, now I'm a senior pediatrician, like many other cardiothoracic surgeons, cardiologists, pulmonologists, rheumatologists. I have metamorphosized into a into a COVIDologist also. Many doctors have written to me to ask me, how long will the immunity last? They are very concerned. These questions I am trying to answer because I will be dwelling into basic immunology today behind this, and that will, to a certain way, answer this question also. Many of my colleagues, doctors, are really concerned because of the death of many doctors all around India. And the most important thing they often ask is, if I do an antibody test, will I know if I am protected? That's also because I dwell into basic immunology in this, I would be able to answer this question also to a certain extent. Now the evidence which is quoted is a study done in Lancet, article in Lancet in March 6, 2021. Single dose administration and the influence of timing of the booster dose on immunogenicity and efficacy. This is AstraZeneca vaccine. A pooled analysis of 4 randomized controlled trials. Therefore, they are quoting this study. And what does the study say? They say, the authors say, in our study, the vaccine efficacy after the second dose was higher in those with a longer prime booster interval, reaching 81.3% in those with a dosing interval of 12 weeks or more versus 55.1% in those with an interval of less than 6 weeks. Less than 6 weeks, it was 55.1% in this study. Why in the hell were we giving the doses 4 weeks apart? Now, They go on to say in exploratory analysis, a single standard dose of this vaccine, of AstraZeneca vaccine, had an efficacy of 76% against symptomatic COVID-19 in the first 90 days after vaccination, with no significant waning of protection during this period. It's not clear how long the protection may last with a single dose, because follow-up is limited to the time periods described here, and for this reason a second dose of vaccine is recommended. Therefore, when you postpone the dose, you have two worries. Number one, will the first dose immunity last till the second dose? They say yes, it lasts. There is evidence. It lasts up to 90 days. And will the second dose be better or at least equal to the if the dose was given after 4 weeks or 8 weeks? They say definitely the booster is higher. The authors come to the conclusion that exploratory analysis present in this report show protection with dosing intervals from less than 6 weeks to 12 weeks or more, and that a longer interval provides better protection after a booster dose without compromising a protection in a 3-month period before the second dose is administered. It's self-explanatory. But it seems that even in February, an expert panel of WHO recommended an 8 to 12 week interval between 2 doses of AstraZeneca. Clinical trials at that time had shown that the vaccine 4 to 6 weeks apart had around 54% efficacy compared to 79%. The gap was 12 weeks. It's really surprising that the health workers all over India continue to receive CoviShield with a gap of 4 weeks, even in March, April, etc. Whenever I hear discussions on COVID vaccine recently in the both television and other programs, I'm really surprised that the experts discuss it as if it's the first time a vaccine has been discovered, and we don't know how vaccines act before. We have been giving it for more than 40, 50 years to children, and a lot of basic science goes into that. Now, I would discuss basic immunology of vaccine because it answers many of the questions which the doctors and the public have. Don't worry about it. You will be able to grasp, even if you are not a health personnel. I will put it across to you in such a way that about 80% at least of what I say, you will clearly have an idea. You will grasp them definitely. We have two types of immunity. One is the innate immunity, which is present all the time, and it is non-specific, and we have an adaptive immunity, which kicks in whenever we are challenged, either by a bacteria, virus, or sometimes by the immunization. Now, the natural immunity or the innate immunity is the epithelial cells. Your skin form a barrier, the epithelial cells. Your WBC, white blood corpuscles in the blood, like macrophages in them, and also they phagocyte the killer and they provide some protection. This comes in within hours of the whenever you are provoked. Now, this I would consider is something like your security which you have in your flats. They are not armed, they are not very powerful, but they are able to react within few hours. Now, the other one is adaptive. Adaptive is when you are attacked by a bacteria, virus, or when you are given immunization. It contains of two types. One is produced by the B lymphocytes, which produce plasma cells and then the antibodies. These can be measured. The other is T lymphocytes, which produce cytokines and produce killer cells, which kill the cells which contain the virus. It's more important for intracellular pathogens. Now, both these things, T lymphocytes and B lymphocytes, takes days, a few days to react, but they are definitely more specific, more powerful. Among T lymphocytic reaction and B lymphocytic reaction, T lymphocytic reaction is more effective and more long-lasting, and T lymphocytes are required for B lymphocytes also. But what we measure is the antibodies. Therefore, we are able to measure only B lymphocytic reaction in day-to-day practice. Now, one slide about T lymphocytes, which are supposed to be more important, but we will not be talking any further because we cannot measure. Now, whenever the pathogen comes in, and this is the kicks off the macrophages, it stimulates the T lymphocytes. There are two types, CD4 and CD8, and they produce what is known as cytotoxic cells, killer cells. And these killer cells directly kill the bacteria outside, and more important, if they are intracellular, they kill the whole cell directly or by producing cytokines, which are called tumor necrosis factor, etc. Now, we will not be discussing this any longer. If you feel that this is something complicated, forget it. Even if you don't understand all this, you will follow what is coming next. Now, whatever I am trying to tell you is from a classical textbook called In Vaccines by Plotkins, which is one of the well accepted textbooks of vaccinology alone, which many even pediatricians would have not read it in complete. Now, let me take you through simple terms. When a nurse gives you an injection, your antigen, what she gives you is an antigen of the virus inside your muscle. The muscular tissue, the antigen is immediately recognized as an enemy, and in comes the neutrophils, the macrophages. They modify the antigen in such a way they can challenge it and send it through the lymphatics to the lymph node. They take it to the lymph node. The lymph node are small, present in your axilla, where they have, if the injection is given in the arm, they are trapped there. Some of them travel to the spleen also. Now your antigen has reached the spleen or the lymph nodes. The lymph nodes are normal nodular structures which are present in the axilla and inguinal region. Now the moment it reaches here, there is the B cells ready for it. But what happens is, if you look at it, all this is taken from this textbook, and in response to protein antigen reaching lymph nodes or spleen, the B cells capable of binding to this antigen with their surface immunoglobulin. Their surface immunoglobulin, they react, they immediately bind to this, and what happens is undergo brisk activation in the extrafollicular reaction. B cells rapidly differentiate into plasma cells. These are plasma cells that produce low affinity antibodies of the immunoglobulin IgG, IgM, etc. And they appear at low levels in the serum within a few days. So one step is the antigen comes here, the B cells immediately attach to the antigen, they convert into plasma cells, they produce low affinity antigens, and within a few days there is a low level of serum antibody. They have low affinity to that particular antigen. For example, if it's Covid-19, they have low affinity, low levels, but within a few days they are there. Then something else happened. These antigen helper cells, T helper cells, actually we said that T cells are required for B cell action also. The T helper cells that have been activated by antigen bearing dendritic cells trigger some antigen specific B cells to migrate towards the follicular dendritic cells. What happens is the T cells tells these B cell plasma cells to migrate towards the germinal center. The germinal center is a place something like the nucleus in a cell where the dendritic cells are present. Once these B cells migrate inside the germinal cells, something happens. In the G cell, the B cell receive additional signals from follicular T cells, and undergo massive clonal proliferation, switch from IgM towards IgG, IgA or IgE, undergo affinity maturation. Now what happens is that once it enters, here is low affinity, less antibodies. Here once it enters the germinal center, more specific immunoglobulins, more affinity, and massive levels are produced, but it takes some time. Therefore, they differentiate into plasma cells secreting large amount of antigen specific antibodies. This is what happens. And at the end of the day, what happens is a few of these antibodies, at the end of the GC reaction, a few plasma cells exit the nodes and spleens, and migrate to survival niches mostly located in the bone marrow. Some of these plasma cells, they go inside the bone marrow and survive there as memory cells, where they survive through signals provided by supporting stromal cells. They survive in the bone marrow. Now let's have a general look. If you're a little confused, I will give a full explanation. The moment the antigen enters your lymph node or spleen after the nurse has given the injection, recognized by the B cells, transformed into plasma cells, produce low-level antibodies, low-level, low affinity, less amount immediately. Now the T cell help them to migrate inside the germinal center. There they undergo massive production of antibodies through the plasma cells. They are more specific to the antigen, more affinity to the antigen. They are produced in massive amounts. It takes a little longer for this. Some of the plasma cells go inside the bone marrow and survive there. Now this is something which happens, it's called the B cell immunity, protein. Now along with the antibody, simultaneously there is generating of B cell memory response. That's what we would like to see in this slide. Now as we have seen, the antigen enters the spleen or the liver, the memory B cells are generated in response to T cell dependent antigens. The antigens come here, the B cells are stimulated, they produce plasma cells also. The T helper cells direct them towards the germinal center. Here in parallel, see they have entered here, and in parallel to plasma cells, memory B cells are also produced. Notice this is produced simultaneously. At the exit from the germinal center, these B cells do not differentiate into antibody secreting plasma cells, but to memory B cells. Two things happen that transiently migrate through the blood towards the extrafollicular areas of spleen and nodes. They go back into the extrafollicular area or the spleen and lymph nodes, and they persist there as resting cells until re-exposed to specific antigen. Therefore, once the memory B cells are formed, they in the extrafollicular phase, they repair them. Therefore, once you are given a vaccination, along with antibody production in the primary response, memory B cells are also produced, which are there in the extrafollicular space. They wait there. And when the antigen comes again, maybe the second dose of the vaccine or the virus, the memory B cells readily proliferate and differentiate into plasma cells, secreting large amounts of high affinity antibodies that may be detected in the serum within a few days. Therefore, they secrete large amount of antibodies which are there in the body. Therefore, the difference between the primary response and the secondary response is that the memory B cells, when they are present, because you are already immunized, immediately this memory B cell recognize the virus. They start producing plasma cells and they produce large amount of high affinity antibodies because they don't have to re-enter the germinal center. Faster, larger amount of antibodies, more affinity. That is secondary immune response because memory B cells are produced in the first, when you have given the first dose of the vaccination. All along, we have been talking about primary immune response and secondary immune response. We would like to see the antibody titers in various phases of the vaccine response. Then we will get an idea of what these mean and why are we interested in evoking a secondary immune response by giving a booster dose of the vaccination. Now, again, we are taking this from the same textbook of vaccines. And the initial antigen exposure elicits an extracellular response, the result in rapid appearance of low IgG antibody titers as B cells proliferate in germinal centers and differentiate into plasma cells, and IgG antibody titers increase up to a peak level. We have already seen this. What you have to notice, it takes about 10 days to rise, and it peaks at about 30 days in the primary response. 30 days is 4 weeks. Usually reached at 4 weeks after immunization, the short lifespan of these plasma cells results in rapid decline of antibody titers, which eventually returns to baseline levels. Therefore, it peaks 4 weeks. The peak is very narrow. It immediately returns. Now, it shows a precipitous fall, but no doubt up to here they are given the dates, but when does it touch the baseline, they have not given. But they say return to baseline fast. There's a problem here. But this is what happens in a primary immune response. Now, in a secondary immune response is what happens in the second dose. Booster exposure to antigen reactivates the immune memory cells, and therefore there is a rapid rise. Within 7 days it rises, and IgG short-lived plasma cells maintain peak antibody level during a few weeks. There the peak is not so narrow, it remains a few weeks, after which the serum antibody titers decline initially with the same rapid kinetics as following primary immunization. Remember, after the first secondary immune also they fall like that. Maybe here they have written 6 months, maybe more slowly, but definitely they do fall. Now, long-lived plasma cells that have reached survival niche in the bone marrow continue to produce antigen-specific antibodies, which then decline with slower kinetics. Therefore, we said that some of the plasma cells migrate towards the bone marrow. They start producing, the secondary immune response is elicited, they start producing antibodies. So much so, instead of the antibody level dropping straight, they live for a longer time. Therefore, what it means is, therefore, the primary response, slower time, peak is narrow, falls rapidly. If you are able to produce a secondary immune response within a few days, within 7 days it peaks, the peak is also a little broader. And if you have elicited a secondary immune response, we will come to that, it stays. The memory B cells the surviving cells in the bone marrow start producing antibodies, and therefore the antibodies remain for a longer time, provided you have produced a secondary immune response. When you are giving 2 doses, a minimum of 4 weeks interval between the primary doses avoids competition with successive waves. That means if you give 2 doses, at least 4 weeks should be there gap between the 2 kill vaccines. Otherwise, the peak antibody level will interfere. Now, this 4-week interval is producing 2 primary immune responses. This we give it in children because the maternal antibodies are there, the children's immune system is not fully developed. Therefore, in fact, we give 3 doses to make sure there is a primary immune response, and maybe we will give it 1.5 years dose to produce a secondary immune response. Now, if you want to produce a secondary immune response, a 8-week interval of the doses are maintained between 2 kill vaccine for secondary response. If you want to produce a secondary response which has a rapid rise, a longer peak, and a fall, then a minimum of 8 weeks should be maintained. For all of us health workers who have taken the doses 4 weeks apart, we have only produces 2 primary immune response, and the antibody level is not likely to last long. You may ask me, why did they advise this? Because we were in the midst of a pandemic. They wanted to produce rapid rise of antibodies. They probably thought that 4 weeks is enough. For even 6 weeks is not enough to ensure a secondary immune response. Now, since the affinity maturation B cells in the germinal centers and formation of memory B cells take at least 4 to 6 months, the schedule of 0, 6 weeks, and 6 months quite well fulfills this requirement. I'll explain it further. If you want to produce a secondary immune response, this says minimum of 4 to 6 months interval should be there. But many other literature suggest 8 weeks is enough. For 8 weeks interval, that's what the Britishers have taken up. It will produce memory cells will be there, a rapid rise, a longer peak, and it will fall. But the formation of cells in the plasma cells in the bone marrow, producing cells this 6 to 8 for antibody level will not be there. Therefore, the plasma cells in the bone marrow wouldn't have produced. the antibody levels. Therefore, you will get a primary and a secondary response with a 8-week interval. Therefore, what should we do if we have taken especially doses 4 weeks or 6 weeks apart? The best thing to do is after 6 months take a booster if it is available. That is what the immunologically that will be the right thing to do. Science is not straightforward as that, and I might contradict what I said already to a certain extent. Now, generally, more than one dose is needed for killed vaccines for better induction and recruitment of more number of germinal centers. Therefore, even one dose, you might be able to, the B cells will produce the memory B cells, and within a period of say 8 weeks or 6 months, the memory B cell should be ready inside the extrafollicular space. Thereby, one might argue a single dose is enough. The second, when the virus comes, it produces a secondary immune response. But generally, that is not the way. In killed vaccines, we always give more than, we give a booster dose also. In live vaccine, the virus stays in the body for a longer time, producing the primary and the secondary immune response. Now, you might argue that things have become a little different with modern vaccines. For example, in Johnson & Johnson vaccine, which is a DNA vaccine, given in a vector, a single dose is given. Now, it is not a live vaccine which multiplies, but even then a single dose is given. Probably the argument would be that it's inside the adenovirus. The adenovirus stays longer, though it doesn't multiply, releases the DNA over a slightly prolonged period. Then that must be applicable to AstraZeneca vaccine also, because it's also a vector vaccine. But whatever it is, this is not very clear from whatever I could read in basic immunology. And I would still consider these vaccines as killed vaccine, and a booster dose 8 weeks apart is a must. Let's now find out, with all this discussion, have we found the answers for the questions we asked at the beginning? Why is India increasing the dosage to 12 to 16 weeks for CoviShield? Because an exploratory analysis, a single standard dose of this AstraZeneca vaccine had efficacy of 76% for symptomatic Covid-19, and it lasted for 90 days. Between the first and second dose, even if you give it for after 16 weeks, the effect lasts. And the vaccine efficacy for the second dose was higher than those of the prime dose, 81.3% versus 55.1%. Therefore, this is straightforward. We are fully justified. But there is a small issue here, which, at the end of the day, the government always doesn't say the whole truth. The University of Oxford sponsored studies were initially planned as single dose studies, but were amended to incorporate a second dose after review of phase 1 immunogenicity data, which showed a substantial increase in neutralizing antibody with a second dose. First, they thought that they will have only one dose for AstraZeneca. Then they decided for second dose. After initially providing consent to participate in a single dose study, some participants chose not to receive the second dose, provided a self-selected cohort of single dose recipients. Because those people who did not agree for a second dose, they provided the base for studying how long the one dose people last, one dose antibody level last. Additionally, because of the time required to manufacture the second dose, there were delays in administration of the second dose for a larger number of trial participants who received the 2-dose schedule. Because suddenly when they said the 2-dose schedule, the vaccines were not ready, and therefore some people started receiving it after 5 weeks, some after 6 weeks, some after 12 weeks, and few of them after 12 weeks. Now the problem here is, our people this is not the way a proper study is done. This is an accidental finding which a pure statistician will not agree to, number one. Number two, real life evidence of immunogenicity are not present beyond 12 weeks. Beyond 12 weeks, they have done some study using the efficacy they have looked at by looking modeling. Therefore, there is some issue there. The real life evidence is there up to 12 weeks, not up to 16 weeks. And the real life evidence which the Indian government is talking about is also not will not be agreed upon by a Puritan statistician. That's, by the way, I'm pointing. I'm sure you're waiting to hear what explanation I can give for UK shortening the dosage to 8 weeks when India made it to 12, prolonged it to 12 weeks. Now, the 8-week dose should produce a higher earlier peak and secondary response. What UK is aiming as to peak, because of the new strains from India, they have panicked a little. They want to produce 2 peaks like this and the secondary peak earlier. The secondary peak, therefore, they get a higher immune response at the 8th week itself or the 10th week itself. They are worried that what is a protective titer correlate is not known for certain. But when they are giving 8 weeks apart, they are producing a higher peak earlier. And of course, there is 8 weeks may be enough to produce a secondary immune response. While India, on the other hand, relies on the UK data, which shows that up to 90 days, the first dose, the antibodies remain high and do not wane. Therefore, it's a question of India depending on UK data and United Kingdom panicking and relying on basic immunology to reduce the dosage to 8 weeks, not based on real life data. Now, the other tricky question is, why is Covaxin being given 4 weeks apart? Now, why? That's only data available. The study done in Covaxin was done by giving 2 vaccines 4 weeks apart. Why did they do it? Because they wanted to produce 2 peaks as early as possible. Probably that was the reason. But if they had given 8 weeks apart, definitely this will produce 2 early peaks but not prolonged antibody level. Definitely 4 weeks apart will not produce the secondary immune response. It will not produce. It will produce only 2 primary peaks. And then why does it, are the scientists not knowing this? But there's a medical legal aspects in fact. The matter is, the study has been done using 4 weeks apart, and now nobody will stick their neck out and say, no, you give it 8 weeks apart. If I am in private practice, a patient comes to me and asks me, and I trust him, I would say, okay, go ahead and do the 8 weeks. These are the facts. It's better to take 8 weeks apart because you will have a secondary immune response and the antibody level will last longer. But the central government, when they have not done the study, will not be able to do it. Therefore, it's a question whether you trust the basic immunology or you just rely on the field study you have done. Now, the other question is how long the immunity lasts. Especially the people who are most worried about this are doctors, with more and more doctors succumbing to COVID now with the secondary wave. Now, theoretically, even a 8-week interval should produce a prolonged antibody level, as produces a secondary response, while a 4-week interval in Covaxin will not, because it does not produce a secondary immune response, which we have seen. Therefore, now with the advice that 12-week interval or 12 to 16 week, 16 week is not very much on scientific basis, but if you give a 12-week interval, definitely you should be producing a long-lasting immunity. That's what basic immunology tells us. But surprisingly, most of the experts who have come on television and talked, they are very skeptical. They say we require real life data. Nowadays, people don't trust basic immunology. They trust only on real life data. And many of them are telling it might not last more than 6 to 8 months. I don't know why they are pessimistic about it. It's better to be pessimistic about COVID-19 because it proves us wrong. Whenever we were optimistic about COVID-19, it has proved us to be wrong. The last issue in question is the antibody level. Now, many doctors ask me whether they can take the antibody level to check whether the vaccine has been effective. But the real advice is that a low antibody level does not mean T-cell immunity is not functional. Therefore, it is not advisable. That's what the government says, and that's what most experts would advise you. If you take a antibody level and it comes low, it doesn't mean that you are not protected. And further now, with no vaccines available to give another dose, it is better that you don't do it. But there might be a reason for doing it. The reason is a high antibody titer means the memory cells are also stimulated. I will tell you what it is. See, whenever we have seen the cells, simultaneously in the germinal center, the plasma cells are stimulated to produce high levels of antibody. Same time, in the germinal center, same time, the memory B cells are also stimulated, which produces long-time memory. Therefore, if you measure this and you find out the antibody level is very high of protective antibodies, it is very likely that the bone marrow, memory B cells are also will be very high. There is a correlation between this. This is given in standard immunological books. Therefore, if you take the second dose and after 2 weeks you do an antibody level and it comes in thousands, you can rest be assured that your long-term immunity is also probably ensured. Now, the other question is, we measure anti-spike antibodies. Does it mean that it is is it immune correlate? But is it not better to measure neutralizing antibodies, which are not done in the laboratory? Generally, yes, the anti-spike antibodies are generally considered. That's all we can do. It's considered as immunological correlate, though there may not be absolute evidence for that. Now, more and more mutants are coming about. You get everyday report telling that the vaccines are effective but less effective. And what do they say? The neutralizing titer at which it is effective is much more. The neutralizing antibody is less sensitive to these vaccines. Now, again, this is most of the virologists will tell you that when we look at the protective correlate, suppose you require 1/8 dilution of the neutralizing antibody for your vaccine to be effective, they keep on doing in much higher dilution, 1/8, 1/64, 1/256, etc. Therefore, normal viruses, normal clones of the viruses may be neutralized by even 1/256, while the new mutant strain may be neutralized only by 1/512. But even that is enough for protection. It's much above the protective level. Therefore, generally, the opinion of the virologists is most of the mutant strains are susceptible to our vaccination. In conclusion, it is well accepted when there is a low supply of vaccine, a policy of initially vaccinating a larger cohort without compromising their protection with a single dose might provide better overall population protection than vaccinating half the number of individuals with 2 doses in short term. Further, the Indian government is not planning to give only single dose. They are only postponing the second dose to 12 to 16 weeks. I think it is a right decision and they should be lauded for that decision. With this, we come to the end of the presentation. If you have got the appetite, time and inclination to watch more videos, you can go to the site where we have put nearly 24 educational videos, mainly touching pediatric topics. There are some blogs on this particular link also. So thank you for watching and goodbye for the time being.